pregnancy. So in this session I will be dealing about fertilization and implantation, how the placenta is formed and its function, then physiological changes during pregnancy. Fertilization. So the process of union of sperm with egg, and it will result in the formation of zygote. This whole process, it happens at the ampulla of the fallopian tube. Ampulla, it is the widest and the longest part of the fallopian tube. Here there is transport of gametes, that is sperm and the ovum, and there is sperm capacitation and there is gamete fusion. Now we can look how the ovum is transported. So at the time of ovulation, the fimbria of the infundibulum, it will encircle the ovary and it will rub it, and it picks up the ovum from the peritoneal cavity. This ovum will be directed towards the uterus by the continuous beating of cilia and the contraction of smooth muscles of the fallopian tube. So by the beating of movement of cilia and the contraction of smooth muscles of the fallopian tube, this ovum will be directed towards the uterus. The viability of ovum is 6 to 24 hours. So if fertilization is occurring in this period, the pregnancy will occur. If fertilization doesn't occur, the ovum will die out and it will degenerate. Now, the transport of sperm. 200 millions of sperms is produced per ejaculation, and it is deposited in the vagina. 50 to 100 sperms will only reach the ovum and only one will penetrate it. The viability of the sperm after ejaculation is hardly 1 or 2 days, that is 24 to 48 hours. So now what is sperm capacitation? The activation of the sperm inside the female genital tract is called the sperm capacitation. It will take 1 to 10 hours for capacitation, and the cholesterol content of the acrosomal membrane, it decreases and it becomes weak. So there is easy release of enzyme from the sperm's head, and the sperm will become permeable to calcium ion. So the calcium influx will be there, and the flagellar movement of sperm become whip-like movement, and the enzymes will be released from the acrosome. That is, there will be hyperactivation of sperm, and this will enable the ovum to fertilize. So the fusion of gametes. So there are 4 steps. That is chemoattraction, penetration of sperm through the penetration of sperm through the ovum covering, fusion of sperm with the oocyte, and ovum activation. So what are the steps in fertilization? There will be chemoattraction of sperm to ovum and adherence of sperm to zona pellucida, penetration of the zona pellucida and the acrosomal reaction, adherence of sperm head to cell membrane of ovum, and breakdown of the area of fusion and the release of sperm nucleus. Now the fusion of gametes. So there will be chemoattraction, that is attraction of sperm to ovum by substance produced by the ovum. Then penetration of sperm through the ovum covering. There is a penetration of corona radiata and zona pellucida by the proteolytic enzymes. That means it should cross cells of corona radiata, then the zona pellucida layer, then the perivitelline space, and after crossing vitelline membrane, it enters. Here you can see the acrosome is releasing. So it will release hyaluronidase and certain proteolytic enzymes, then fertilin, which is a protein on sperm's head which will facilitate the fusion. Side, rather than the tip of the sperm's head, the side gets fused. Head separate from the middle piece and tail and swell to form the male pronucleus. In ovum, the second meiotic division is completed. and the chromosome will assume the shape of a nucleus, that is female pronucleus. Each pronuclei has 23 chromosomes which align to form complete complement of 46 chromosomes in fertilized ovum. Okay, so this is the overall view where when the sperm is entering, there will be the corona radiata will be dispersed, and when the sperm will enter the cytoplasm, formation of male nucleus and male pronucleus and female pronucleus. So after this, the fusion of sperm and the oocyte will happen. The equatorial region of sperm will fuse with the vitelline membrane of oocyte. So fertilin, it is an acrosomal protein. It will interact with the protein present on the vitelline membrane, and the genetic material, it will enter the oocyte. It will lead to fertilization, and it leads to zygote formation. The ovum activation will happen at ambula, that is only one sperm it can enter into the oocyte. The further entry of sperm is prevented by ovum activation, that is polyspermation block. So release of calcium from egg reserve to perivitelline space. The membrane potential of ovum decreases. So how the polyspermation is blocked, that is entry of more than one sperm is blocked. That is by blocking zona pellucida and by blocking the vitelline membrane. So after fertilization, diploid chromosome number is restored. Second meiotic division will happen and the second polar body will be extruded. At this point, determination of sex takes place. The fertilized ovum, it begins to divide. And after this fertilization, the next main event is implantation. That is the attachment of the fertilized ovum to the uterus endometrium. The most common site is the dorsal wall of uterus. So the blastocyst must form, then the transportation of blastocyst and the implantation of blastocyst and the decidual reaction will happen. So how the blastocyst is formed? So the fertilized ovum is a zygote. It divides and divides and divides and forms a morula, that is a 16-celled stage. And on further division, it forms the blastocyst. So this is a blastocyst. Here you can see it is arranged as trophoblast and in outer trophoblast and inner cell mass. Actually, the inner cell mass surrounded by a layer of trophoblast. This inner cell mass will be converted into embryo, whereas the trophoblast cell layer has a sticking property. Outer is a syncytial trophoblast and inner is a cytotrophoblast. This trophoblast is covered by zona pellucida, and this zona pellucida prevent the implantation of zygote in the fallopian tube. Now the blastocyst has to be transported. For next 3 to 5 days, this happens by fluid current, by the ciliary movement of the fallopian tube and by the ciliary movement of uterus. Then there will be weak contraction of the fallopian tube. This blastocyst, it floats in the uterine cavity and the zona pellucida will disappear. When the zona pellucida disappear, it becomes sticky and there will be implantation of the blastocyst. That is, the syncytiotrophoblast will secrete proteolytic enzymes. It digests and liquefy the endometrial cells. The blastocyst erode and burrows the endometrium. This is known as the endometrial implantation. So implantation normally occurs 8 to 9 days after fertilization. Now implantation. So from the ovulation when we are taking, it occurs 7 days from ovulation. This zona dissolved by enzymatic action. The blastocyst burrows into endometrium. Usual site is the upper part of wall of uterus. Placenta develop at this site. This is the schematic diagram. There is here you can see the blastocyst is burrowing into the endometrium, and there is a fibrin deposit around it, and after that the uterine epithelium is reformed. So this is the overall happening from the ovulation, fertilization and the implantation up to implantation. Here you can see the amniotic cavity and the trophoblastic cells is invading the endometrium. If the pregnancy is occurring, the ectopic pregnancy, it is a pregnancy. If the pregnancy occur outside the uterine cavity, it is termed ectopic pregnancy. For example, the fallopian tube, ovary, abdominal cavity, it is known as ectopic pregnancy. So, early nutrition of embryo. Before implantation, there will be endometrial secretion. So, first week following implantation, decida will be supplying the nutrients. After 1 week, only the placenta will be supplying the nutrition. So, before implantation, it is an endometrial secretion. The first week following implantation, it is a decidua, and after 1 week, it is a placenta. The decidual reaction. So, after implantation, the endometrium is known as decidua. Endometrial cells become enlarged, vacuolated, filled with glycogen and lipids, and these cells now are called decidual cells. It will provide nutrition to the growing fetus until the placenta is formed. So, till the formation of placenta, this decidua will provide nutrients. Now, the corpus luteum of pregnancy. So, the corpus luteum, it is formed during the post-ovulatory period. That means after ovulation, and it usually regresses after 24 days. In pregnancy, it will persist up to 3rd month. So, it will maintain, that means it maintains pregnancy up to 3rd month. It is a major source of estrogen and progesterone. After that, the placenta will take over the function. The placenta will secrete the hormones. So, going on to the placenta, it is a temporary endocrine organ of pregnancy. It is a link between maternal decidua and fetal chorionic villi. It is connected to fetus by the umbilical cord. It formed during 6 to 12 weeks. It is a disc shaped, and the weight is 500 grams and the diameter is 15 to 20 centimeter. This is a pic of placenta. Here you can see the fetal surface of the placenta showing attachment of the umbilical cord with a ramification of the umbilical vessels. That is, umbilical vessel rami can be seen. This is a maternal surface of the placenta showing shaggy look with a cotyledon limited by fissures. Now, how the placenta is formed. So, after implantation, the endometrium is known as decidua. The decidua basalis, it is the part where the placenta is developed. That is, in the image you can see decidua basalis. It is a part where the placenta is developed. Now, decidua capsularis, it separates the embryo from the uterine cavity, and the decidua parietalis part lining rest of the uterine cavity. So, rest of the uterine cavity is decidua parietalis. So, the part of decidua basalis means the part of the endometrium where the placenta is developed, and decidua capsularis, it separates the embryo from the uterine cavity, and the rest of the uterine cavity is decidua parietalis. So, in this decidua basalis, the maternal blood vessels proliferate. The syncytial trophoblast send codes into decidua basalis, and the placental villi will be formed. In the placental villi, blood capillaries grow from the vascular system of the embryo. The fetal blood circulates in blood vessels in substance of villi. The maternal blood will circulate in intervillous space, and the exchange occur through the wall of villi. So this is a villus, and here you can see the fetal blood capillary, and the maternal blood is present in the intervillous space. The placental villi are surrounded by blood-filled maternal sinus, that is intervillous space. The maternal blood flow through the uterine arteries into maternal sinus, and after diffusion, it go back by uterine vein. Fetal blood flow through the umbilical arteries and went back by umbilical veins. Now the placental membrane. So maternal and fetal blood are separated by placental membrane. So basically there are four layers. That is the endothelium of the fetal blood vessels and the basement membrane, mesenchymal tissue, cytotrophoblast and the basement membrane, and the syncytial trophoblast. So here the transport of oxygen, nutrients, waste product occur between this membrane. So there is endothelium. In this diagram you can see the endothelium of blood vessels, then the connective tissue, cytotrophoblast, syncytial trophoblast, and maternal blood. The blood will be flowing through two umbilical arteries, then through the villus capillary, through the single umbilical vein in fetus. The uterine artery, from the uterine artery to the maternal sinus and to the uterine vein in mother, in the case of mother. Now the functions of placenta, it is having many functions: nutritive function, respiratory function, excretory function, protective function, immunoglobulin function, transport of IgG, endocrine functions. So the nutritive function include, it is a major function of placenta. It starts from 4th week. The transport of nutrients from maternal to fetal blood. Glucose by facilitated diffusion. Fatty acids, water, sodium, potassium, chloride, vitamins, and ketone bodies through simple diffusion. Fe2 plus, Ca2 plus, amino acid by active transport. So respiratory functions, that is transport of O2. Dissolved O2 from maternal sinus of placenta, it diffuses into the fetal blood along the pressure gradient. That is, PO2 of maternal blood is 50 millimeters of mercury, whereas the fetal blood is 30 millimeters of mercury, and the hemoglobin of fetus, it is having high affinity for O2. That is higher hemoglobin concentration. That is the oxygen dissociation curve shift to left due to its highest binding capacity. The transport of CO2 from fetus to mother's blood occur by diffusion along the pressure gradient. Then it has excretory function: urea, uric acid, creatinine diffuses to maternal blood along concentration gradient. Then it is having protective functions, that is barriers against bacteria and harmful substance. The placental progesterone decreases uterine contraction. Hormonal secretion help in proper growth of fetus. Then it is having immunological functions, that is it prevent antigenic reaction by keeping mothers and fetal blood separate. Trophoblasts do not have MHC1 and MHC2 genes. No antibodies is produced against the fetal protein. Fetus for that is it is for in the case of mother it is considered as a foreign body and it is not rejected. Then the transport of IgG, that is IgG antibodies transported from mother blood to fetal blood. It provides innate immunity. RH incompatibility is due to IgG antibodies. Then there is endocrine functions, that is syncytial trophoblast will act as an endocrine gland. It secretes HCG, human chorionic gonadotropin, human chorionic somatomyotropin, HCT, that is human chorionic thyrotropin, placental estrogen, placental progesterone, relaxin. In that human chorionic gonadotropin, it is a polypeptide hormone. It is produced soon after fertilization. It can be detected in the maternal blood 6 to 8 days after fertilization. It reaches its peak between 60 to 90 days of gestation. So what are the functions of HCG? It maintains the function of corpus luteum up to 7 weeks. That is, it is having LH-like action. Convert the corpus luteum of menstruation to corpus luteum of pregnancy. Stimulate the fetal testis to secrete testosterone. Development of male genital organs and the descent of testes. Now, human chorionic somatomyotropins. It is also called as human chorionic placental lactation, that is HPL. It resembles a growth hormone. It is a maternal growth hormone of pregnancy. The secretion starts from 5th week of pregnancy. The secretion is proportional to the weight of placenta. Low maternal human HCS indicate placental insufficiency. And what are the functions of HCS? It is having lactogenic action in lower animals, growth stimulating action in humans, decreases insulin sensitivity and decreased glucose utilization by mother, So that more glucose is available to growing fetus. It promotes lipolysis, that increases fatty acid concentration in mother, and it will help in retention of electrolyte. Estradiol is a major estrogen during pregnancy. Estradiol is the most potent one. It is synthesized by the corpus luteum and syncytial trophoblast. Functions: it growth and development of maternal reproductive organs, that is uterus and breast. Development of lactiferous ductal system in mammary gland. It relaxes the pelvic ligaments and allow easy passage of fetus through the birth canal. Now the progesterone. It preserves the pregnancy by promoting growth of endometrium. It decreases the contraction of pregnant uterus. It helps the estrogen to prepare mother's breast for lactation. Immunosuppressive role to protect the fetus. It acts as a precursor for corticosteroid synthesis by fetal adrenal cortex, helping the growth and development of fetus. Now there is a hormone which is called a polypeptide hormone relaxin. It is secreted from the corpus luteum, uterus, placenta and the mammary gland. It relaxes the pubic symphysis and the pelvic joints. It softens and dilates uterine cervix and facilitates delivery. Now the feto-placental unit. The fetus, placenta and the mother interact in the formation of steroid hormones. So placenta can synthesize pregnenolone and progesterone from cholesterol. Some of the progesterone enter the fetal circulation and leads to the formation of cortisol and corticosterone, that is from fetal adrenal gland. So the pregnenolone, it is converted into DHEAS, that is dehydroepiandrosterone sulfate, and 16-hydroxy DHEAS. So these two are transported back to the placenta and they are converted to estradiol and estriol. Urinary estriol of the mother is an index of state of fetus. So this is the overall view. Then there is something called as pregnancy test. So based on detection of HCG in urine, we can detect within 14 days of conception. So in serum, within 7 days of conception. So here you can note if 2 lines appear, the biological test is outdated. Here you can see if 2 lines appear, test is positive. In that we will have a sample well in which we add a 3 drops. Then there is something called as immunological test that is based on antigen antibody reaction. So one is a Grav index test, that is agglutination inhibition test. That means suspected urine. That is urine which is containing the HCG is mixed with the HCG antiserum. So then it is mixed with the HCG coated sheep's RBC, that is latex particle. So if there is no agglutination, the pregnancy is positive. If there is agglutination, pregnancy is negative. If the urine contains HCG agglutinated by the antibodies of antiserum, all antibodies are used. So no free antibodies are available. The latex particles are not agglutinated and the agglutination is absent, so the pregnancy test is positive. So if the urine doesn't contain HCG, reaction between HCG and coated RPC or latex particle and antibody in the serum occurs. So the agglutination is present, that means pregnancy is negative. Now there is another method, that is direct lattice agglutination test. Here latex particle coated with anti-HCG antibody mixed with urine. So if the urine is with HCG, the agglutination occurs and the pregnancy is positive, test is positive. And if the urine is without HCG, there will not be any agglutination and the pregnancy will be test will be negative. The third method is ELISA, that is enzyme linked immunosorbent assay. Here 2 antibodies are used. The monoclonal antibody that bind HCG, antibody linked with alkaline phosphatase to sandwich HCG, detected by color change after binding, and this is more sensitive and more specific. So there are other tests which are used, that is immunofluorometric assay, radioimmunoassay using iodine 125. It is highly sensitive. Then there is another by USG, detect pregnancy by 29 to 35 days of gestation. Gestational sac by appears by 5 weeks. Fetal pool and cardiac activity is by 6 weeks.